Hey everybody. So my name is Ethan Short. A few things about me. I'm a Lane Community College student. This is actually my last term here, so a little bit sad. Um, let's hop right into first question. So favorite movies or books? I really like the Lord of the Rings series. Those have to be my favorite books. Favorite movie is a lot tougher for me, but I'll settle on Die Hard. It's not a Christmas movie, by the way. It's just themed around Christmas, but that'll bring up a lot of stuff. So, moving on. So, here's my DNA model to answer the first question. It's pretty simple. Use post-it notes. See the nitrogen bases in different colors. I don't know if you can see the labeling. That looks like it's kind of getting blurred out a little bit, but I have like a note up there. Um, Obviously, this is flattened out, right? A normal DNA strand is going to be twisted. That sugar phosphate backbone is going to go around and around. Then you have your major grooves, minor grooves. Side note, so I'm a computer scientist, developing software. I'm a software engineer. So I actually made a quick program to basically kind of do this on by itself. So I just want to show you real quick because I think it's pretty cool. Yeah, so here it is. I just call it my DNA finder. I think it's pretty cool. I don't expect you to know Python code at all. That's totally fine. I'll just show you what it does. So you just enter all your key values here, T, A, C, or G. Um, and then it's going to convert the sister string or the string to go along with it. So I'm just going to go ahead and run the file. Oops, sorry, it just takes me a minute. Okay, there we go. So you can see the visual up on the screen, and it's actually drawing the complementary DNA strand, which coincidentally is the exact same as this one right here, which is pretty cool. This is another way to learn. It helps me cement these topics just a little bit better. So moving on to meiosis now. So let me just get that up for you guys. Yep. So here it is. I'll zoom out all the way so you guys can just see it like that. We see the pre-stage, meiosis one, then meiosis two. So let me just zoom in a bit and go through this real quick. Okay, so basically we have G1 and G2 right here. We're just getting set up with one single cell, guys. We're just starting with one cell. So we have two pairs of chromosomes. They're going to go in a cross-like pattern. Crossing over has not happened yet, but we're getting centrioles in the cell as well. So moving on, starting with meiosis one here, which is the start of prophase one. So we can see the cell membrane still here, but the centrioles are starting to send out their spindle fibers, and we can see crossing over does occur. I will define crossing over at the end here. So moving on to metaphase, they line on that midline, or the centrioles align on the center of the cell, and they start to grab those genes. So anaphase is when the cell starts to stretch and get longer and longer. And then the spindle fibers actually are sent out the other way now. So, excuse me. Um, when you see crossing over still in effect, but they each grab a side, and then boom, they're two separate cells now. Now, just going through this quickly, centrioles are re-added to the two separate cells now. Well, just two, one hand. Um, And then prophase is when those centrioles realign. And then metaphase, we see the spindle fibers going out. We can see it's going to start to grab half of these genes over here. Um, sorry, half of the chromosomes. And then in anaphase, we can see this successfully. The sister chromatids now are separated and now pulled towards the opposite sides of the cell. And then telophase, of course, is now we have four distinct separate cells, and they're actually all different from each other. They're just variations of each other with actual different genetic material, which is pretty cool. So now I'm going to go ahead and define, so crossing over, as I showed you before, happens during meiosis one, during the prophase section, okay? But basically, we're just exchanging segments of DNA here. And as a result, the offspring produced by this is going to actually have genetic variation, which is important because independent assortment also gives us additional genetic variation. And basically, what that means is going to happen during metaphase, which is here. The centrioles can grab any ones they want. They have to grab a little one, have to grab a big one, but they can grab any one they want. So, basically, the combinations are 2 to the nth power. So n represents this set of pairs. So in this case, there are 2 pairs, so there would be 4 possible combinations, um, if that makes sense. So let's move on here, and look at our next question. Okay, so now we're in Unit 2, and we're going to work on some of these definitions. So, first off, what's the difference between genes and alleles? So basically, a gene is just a segment of DNA that has a set of instructions for a protein or function, whereas an allele is a variant of a gene, that can lead to trait variations, basically. So genes are broad units of heredity, whereas alleles are like specific versions of a particular gene, if that makes sense. So moving on, we have phenotypes and genotypes. Basically, a phenotype is just an observable trait, like we'll see in Puff's pedigree. Um, where some of the dragons can breathe fire, some can't. That's a phenotype, and it's an observable characteristic. Whereas a genotype is their actual genes that can lead or possibly lead to that phenotype, as we'll also see in Puff's pedigree and the other pedigrees, we'll look at here in just a couple minutes. But basically, think about it in terms of genotype is like a genetic composition, but a phenotype or a phenotype, is the actual genetic, excuse me, makeup of something. Now we have homozygous and heterozygous, but basically, so homozygous just means something has identical alleles for a gene. So like no genetic variation, basically. But heterozygous means the alleles have different genetic variations for a gene, for a specific gene. Um, yeah, I think that sums it up pretty well. Let's see. Let's see. Sorry, I'm just trying to make this quick. All right, Puff's pedigree. Let's pull that up here. Okay, so I've done this probably a little different than you might have. Just kind of on a board to get my thoughts out there. Kind of what works for me. But basically, as we look at the pedigree, we got the mother side to the left, which can all breathe fire, and then the side to the right, which cannot breathe fire, only starting at Smaug. So basically, what the first question is this trait dominant or recessive? I believe it's dominant because we can see that, actually, no, this is exactly the opposite of what I said, of what I meant. I'm sorry. So it's definitely recessive because we know that it can be inherited without both of the parents having it. So we can see Smaug's mother and Smaug's father, not possessing the trait, but still possessing enough genetic material to actually have that phenotype as a fire breather. Um, basically, so we know that just looking at this, um, Saphira is going to have, um, so little f's basically. So, and then there's going to be 2 of them. with the grandmother and grandfather most likely have a big F and little f, and then another big F and little f, and they transferred that little f down to Smog. So it's going to be little f, little f, little f, little f, and then Puff also has little f, little f. So basically, they're destined to have a fire breather, and if I'm not mistaken, all of their kids will also be fire breathers as well. And then we know their grandparents, at least one of them has a little f, but a max of two of them could be both big Fs as well. Um, so let's switch over here to the genetic problems. Um, okay. And I'm just gonna answer 6 and 10 here. So basically, the question that is 6 here, let me just uh, pull it up so I know what questions I'm answering. Oh, that's not the right one yet. We'll get to that here in a minute. Um, Okay, so for 6 and 10, basically question 6 is talking about peas. Here, let me just make this a little bit bigger. All right, so it's talking about peas, and tall is dominant over dwarf, and basically it's asking if a plant for tall is crossed with one for dwarf. I'm just gonna ask a series of questions. So A is what will the appearance, the phenotype be of F1 plants? Well, they are going to be tall. Um, because it's always going to have that little f, which is the tall gene, no matter what offspring it has. So F2 or B, so it says what will the phenotypes of F2 and what fraction of the offspring will have each phenotype. Um, so for F2, their offsprings or its offsprings is going to have a 75% chance of being tall and a 25% chance of being dwarf because already it's inherited one of those genes, and then it's crossed with a dwarf plant, which gives like a small percentage that's going to end up as a dwarf. So C says what will the phenotypes of and fractions of an F1 plant crossed with a tall parent. So if F1, we know it's going to be tall. If it's crossed with another tall plant, we know it's going to be tall too. Because both genetic makeup is going to be tall. Um, but the offspring's genotypes are going to be big T and then little t. So then D, the last question in section 6. So what will the phenotypes and fractions of an F1 plant crossed with a dwarf plant? So we know F1 plants are always going to be tall. So always going to be tall, and they're dominant over dwarves, and dwarves would be little t. So half are going to be big T little t, and the other half are going to be a small t little t. So moving on to question 10, we're just talking about a two black female mice crossed with a brown male, and it's going over several litters. So you can follow along with the questions as well. I'm just going to go ahead and answer them, but basically it says, what can you determine about the inheritance of black and brown coat color from this data? The data is in the question, but basically the black coat is going to be dominant. And then what are the genotypes of the parents? So the parents' genotypes, so the both females will be big B, little b, and then the male is going to be recessive little b, little b. So let's continue moving on here to the next question. Okay, pedigree analysis. Almost brought up Puff's pedigree again. Okay, so let me get that up there. Okay, here we go. So I'm just going to be looking at this first one. But the first question here is So I can find it. Okay, is it dominant or recessive? So it's a pretty easy one, actually. I believe it's recessive because we can see 2-2 and 2-3 right here don't have the trait, but their kids do. That's a pretty clear indication, generally, if it's recessive or dominant. If it's going to be dominant, I would like to see at least one parent have the trait to be able to pass it down. So, next question was the genotype of 2-2 and 2-3. Okay, so basically, it actually has to be very specific. So the genotype of 3-2 is going to be two A's. The genotype of 3-4 is going to be two A's. So that means that the genotype of 2-2 has to be big A little a, and the genotype of 2-3 also has to be big A little a. So there's I believe, 50-50 chance because it takes, oh, this is a hard one. But a 50-50 chance that it can be either both little a, um, or actually, no, that's not right. It's a 25% chance that it's little a, because the rest of it is either going to be big A, uh, or a like a 50% chance of big A, or little a, big A, respectively. So we know what the parents are, and then um, its parents are either going to be big A or big A, and it's same with 1 and 4 on this second row here. Okay, so moving on. So let's see. Nice. Actually, I think that is it. So, please let me know what you think. Um, it was great showing you guys this, and have a great rest of your night. See you later.